All right, well, let's get started. We're a bit early, so we might have some stragglers coming in late. But today I'd like to talk to you about partial reprogramming. I'm Yuri Deigin, as had a very nice introduction. I am leading YouthBio, so where we are creating gene therapies based on partial reprogramming. And so today just want to talk to you about this paradigm and give you an overview of what it can do. And of course, the problem that we're all collectively trying to solve is aging, and ideally we'd like to slow it down, maybe even reverse it. Well, ideally we'd like to reverse it. And one way that we can do this is with partial reprogramming, because on the cellular level, it was actually able to do these things because moving forward with aging, you see all these hallmarks of aging. This is a pointer. But anyways, I won't use the pointer. Where moving forward with aging, you have all sorts of cellular damage accumulating or other manifestations of aging, but then you can, with reprogramming, reverse all of the cellular hallmarks, ameliorate all of the cellular hallmarks of aging. And so the next challenge is then to take this and accomplish this on a level in vivo, on a level of already formed organism, because within the context of a single cell is great, but we are unfortunately trillions of cells which are already formed, and that's the challenge. And this is what we're doing at YouthBio, and many other companies also are doing with partial reprogramming, trying to harness the rejuvenating power of reprogramming and apply it to already formed organisms like ourselves. And before we dive too deep into the details of partial reprogramming, let me just take a step back and talk about what aging is. And we all have our own definitions, and unfortunately there's no consensus in the field, as Vadim very eloquently presented, but I think we have some observations from nature that we can make that can give us a general idea of what aging is and what aspects of aging we, as already aged organisms, would really like to ameliorate. And let me just start with some observations from nature, which again, some of you already heard from previous talks, but I guess it's very important for us to take a look at the big picture and not just focus on kind of human aging or mouse aging, because in nature there's just so many different modalities of aging, and the variation in lifespans is just huge. As shown here, it's within 1 million times between different species. Some species live just a few days, other species live for a few years, a few thousand years, and even within mammals themselves, as again Vadim mentioned, there's 100 times difference between lifespans. A mouse lives 2 years, a whale lives 200 years. And so the variation is just huge, and which means that aging can be so diverse, and so we have to figure out what aspects of it we really can manipulate. And also the diversity between lifespans is present in even very closely related species. Like here, rockfishes, which is a single genus, some rockfishes live for just like 10 years and others live for 200 years. And it's the same genus. And here we have two species. One of them, the flagfish, actually lives to 18 years, but there's some rockfishes that live even lower, and it's a smooth continuum of lifespans, like 50 different species of rockfishes between 10 years and 200 years. And so this tells us that aging is quite malleable, and it's within the power of evolution, obviously, but very quickly vary the lifespan. So even in closely related species, it can vary so greatly. And even after environmental conditions change, species can adapt very quickly again by changing lifespan on evolutionary timescales, of course, quickly adapting to changes in the environment. And like I mentioned, not only aging is not universal, the patterns of aging also very variable. And so to me, this means that there's no physical law. It's not the second law of thermodynamics about entropy that makes us age. There's something different, something in biology that makes us age. And so I think that's actually good news, because we couldn't do anything about fundamental laws of physics, but we can definitely do something about biology. And to me, it's pretty obvious that aging is under genetic control on species level. Obviously, it's the genome that determines life histories of species and the speed of aging, and consequently how long a given species typically lives. And of course, within individuals of a single species, there's variation, but it's always within the confines of put upon the species by the genome. And also in some species, the speed can change, can vary based on environmental factors. If there's a famine or a drought, that can actually extend species, and I think this is the mechanism behind caloric restriction. That actually is kind of programmed into the genome that if a species needs to adapt to the environment, if there's some adverse environmental conditions, its biology can extend its lifespan. But the next few observations that I'd like to highlight is, to me, they show that aging is not just under genetic control, but also it's within epigenetic control. And so it's within the power of a single organism potentially to adapt to the environment, as we'll see in some social insects. But of course, for our purposes, if we can manipulate epigenetics, potentially we can manipulate aging, because as I show in the next few slides, there's a very strong epigenetic role in aging. And before we get into the epigenetic control of aging, just a few words about epigenetics, what it is, and just broadly speaking, it's control of gene expression. There's different mechanisms within our genome that control which genes get expressed when, in what type of cells, and as a multicellular organism, this is a necessity because we have close to 200 different cell types. All of them have the same DNA, but there's very different types of genes that, combinations of genes that have to be on in skin cell versus, say, a brain cell. Also, epigenetics controls not just on or off, but there's like a volume level on different genes and the expression of genes, and the volume knob can vary with time, and even like a circadian rhythm, like within a 24-hour cycle, genes go up and down in expression levels. And of course, with aging, we have epigenetic aging clocks, methylation clocks, and we know that the expression levels of different genes and transcriptomic clocks, of course, changes with aging in a very similar way between some subsets of genes in a very similar way across all individuals of the same age, which is the basis for having these clocks in the first place. And let me just share a few observations from nature which show that aging and lifespan are actually under epigenetic control, at least in these species. The most clear examples come from social animals, where the animals share identical DNA, but their lifespans can vary by orders of magnitude. So for example, here we have the honeybee, and queen bee lives for years, while a worker bee lives for just a few months, or so weeks in the summer. And so even a more large disparity is observed in ants. The black garden ant is like a record keeper, the black garden ant queen bee that can live up to 30 years, which is like, when you think about it, I think it's mind-boggling that a small insect can live longer than a horse, or like twice as long as a dog. And yet, again, worker ants live just for 1 or 2 years. Another example that to me is even more powerful is the Indian jumping ant, because here it's, even in the context of the same individual, that individual can be epigenetically reprogrammed during its life if the conditions are right. If the queen dies, these gamergates, they're called, they're reprogrammed into breeders, and that extends their lifespan by several times. And so this shows that within the confines of your genome, epigenetics can vary to a large degree your aging. And so again, epigenetic reprogramming, we see this as an example in insects, and then I think we can learn from it and hope that in the context of our biology, we can also use it with partial reprogramming to accomplish similar goals of extending our lifespan. Another example that I really like is the monarch butterfly, because previously we had social insects which have different social roles, so you can kind of say that maybe they have different life history programmed in the genome, and it's just like kind of one direction that's picked when they're being during embryogenesis, and they're kind of stuck in their genetic program. But of course, the other example of the Indian garden ant shows that you can actually change from one life history to another, which has a greatly extended lifespan. But monarch butterfly is a different story, because there's no different social roles. It's just one kind of social role. But depending on the season in which the butterfly is born, that can greatly vary their lifespan. If they're born in the summer, they live for like a month, but if they're born in the winter, or in the fall, I'm sorry, they can live for up to 9 months because they have to survive the winter. They go down to Mexico to overwinter, they breed there, and then they come back. And so this again shows that, you know, epigenetics can greatly vary lifespan, even within the context of a single social role. Those were insects, but even some mammals have a similar epigenetic life history manipulations. The montane vole, it's a small rodent in Montana. They have this life history that if they're born in the spring, like the monarch butterfly, they mature within the same season and die actually within the same year. But if they're born in the fall, and again they have to overwinter, they can pause their development, pause their sexual maturity, and actually they live for much longer because of this. So again, you can see epigenetic modulation of lifespan. And of course, you know, insects and rodents are great, but we're a little selfish. We want to see what's up with our human biology. Do we have some indications that epigenetics also plays an important role in our aging? And at least for myself, I see this in epigenetic clocks, that the answer is that yes, because we have epigenetic clocks that, you know, by now we have a lot of data that it's not even just humans, it's also in like 190 different species of mammals, and you have a lot of conserved elements between the species. A clock built on human CPGs, human kind of data, works in chimps. So you can also just use a human clock and you can tell how old biologically old the chimpanzee is. And so again, this all ties back for me into the very strong role epigenetic plays in our aging. And so not only that, but of course, the epigenetic clock is synchronized across many tissues. In cells that are so different as a neuron and a blood cell, of course, you're born with the neurons that you're going to die with, and blood cells, they divide and replicate on a daily basis, but yet they show, if not the exact same, very similar epigenetic age. And that's true across many different tissues. And so this conserved nature of these epigenetic clocks between tissues, between species, to me highlights or points in the direction, or strongly at least for me, points in the direction of aging being epigenetically controlled. And so you might say, okay, great, you know, aging is under epigenetic control, but can we actually do something about it? Can we change our epigenetics? Is rejuvenation even possible in the context of you know, fully formed organism? And of course, obviously, we know that during reproduction, rejuvenation is absolutely possible. And in nature, we see rejuvenation happening quite frequently. And it just seems that in mammals, it's reserved for reproduction. We see the rejuvenating event being taking place during reproduction for reproductive cells, for gametes. And of course, when we talk about humans, we have our reproductive cells, our eggs, females, not us, but female eggs, they're the same age as the mother. They're 20-year-old cell, 30-year-old cell. So they've been subjected to this aging for these decades, and yet, after fertilization, all the hallmarks of aging are ameliorated, and of course you get a newborn baby with age 0, reset aging. And of course, not only we observe this in humans, in mammals, we observe this in many different species. For example, in yeast, which is a kind of special example because they're unicellular, single-cell organism, which is kind of both a somatic cell and a gamete, but yet we see that if they're dividing asexually, they don't exhibit this rejuvenation process. A mother cell eventually will die, but if they're forced to divide sexually, which usually happens under adverse conditions, but if we can force them in the lab, they do go through this process of rejuvenation. And you can, if you keep doing this, this kind of mother cell never gets old. It can keep dividing and keep getting the kind of new lease on life every time you induce gametogenesis, the sexual reproduction in yeast. But of course, we observe this in many different species. As I mentioned, in mice, we observe that there's damage getting cleared after fertilization. And In nematodes, Cynthia Cannon's team showed a while back that this also happens just before fertilization. And nematodes are a little different because they're self-fertilizing, they're hermaphrodites, so they know when the fertilization is going to occur, so they can clear the damage just before fertilization. And in frogs, in frog oocytes, frog eggs, we also see this process taking place, that there's active rejuvenation happening after fertilization. And of course, Vadim's team, the great paper that he already cited, that showed that epigenetically, we also observed that epigenetic age is diminished and occurs, the minimum occurs soon after fertilization. Not exactly at the moment of fertilization, but there's actually an active process, presumably an active process of the reduction of biological age, which reaches a minimum during gastrulation. So all of this implies that there is active rejuvenation happening during the reproduction process or after fertilization process. And well, let me kind of bring it back to reproduction and tie it to reprogramming and just give a brief historical overview of how we came to know this wonderful process of cellular reprogramming. And then again, I'll try to tie it back to germline rejuvenation. And before reprogramming proved it wrong, there was this Waddington landscape or Waddington dogma that said that cell fate is irreversible. And cells started this kind of stem cell state on top of the landscape, and they roll down their respective paths towards terminal differentiation, where they end up as a skin cell or a brain cell, and they can never go back. And then this dogma was soon proven false. It was formulated in 1940, but already in 1962, there was first evidence that this dogma might be wrong. And John Gurdon provided this evidence by taking a nucleus from a skin cell and putting it back of a frog and putting it back into a frog egg cell and producing a completely healthy new frog, which meant that all the DNA necessary for the all of the cells of the organism were still present in a skin cell. Oh, and also before, in the Waddington dogma times, the thought was that maybe in differentiated cells, DNA is somehow kind of removed from, irrelevant DNA is removed from the cell. So the cell only retains the DNA it needs to be a skin cell, and so it can never go back to being a stem cell because it actually doesn't have the DNA. And John Gurdon proven this wrong. It said, no, DNA is still there. You can, for any cell type is still present, you can take a skin cell and the DNA is there. And unfortunately, there was like a 30-year pause in this area of research, and the next, well, it wasn't even a breakthrough, it was essentially a repeat of the John Gurdon experiment came 30 years later in 1996, when Dolly the sheep, the famous Dolly the sheep, was cloned, and then subsequently there were many other animals that were cloned, and it just repeated this evidence that all of the necessary DNA is still present, and you can take a somatic cell and it can essentially become a gamete, and all of the necessary DNA is still retained. And the next breakthrough that fully refuted the Waddington dogma came in 2006 with Shinya Yamanaka and Takahashi, showing that actually the process is completely reversible. You can take a skin cell, and even within the context of that particular cell, inducing, using some defined factors, which later came to be known as Yamanaka factors, you can induce the cell all the way back into embryonic state, embryonic-like state, pluripotent state, the stem cell at the top of this Waddington landscape that we saw previously. And so basically they showed that the Waddington dogma is completely wrong. You can move pretty much back and forth in any direction, even laterally. If you kind of use the right reprogramming factors, you can go from like a neuron into a skin cell being reprogrammed. And so that discovery sparked talk of the possibility of, like, is it possible then to epigenetically rejuvenate cells? Which for kind of our purposes is what we're all here trying to accomplish, using this paradigm of reprogramming to actually rejuvenate cells. And of course, it was very soon showed that the reprogrammed cells are not only epigenetically rejuvenated, they're also physiologically rejuvenated. And first evidence of this came in 2011 from Jean-Marc Lemaitres' team at INSERM, who showed that the reprogramming process actually rejuvenates cells and ameliorates several hallmarks of aging. And after that, there were actually many other groups that looked at various other manifestations of aging and various other cellular hallmarks of aging. And this diagram just kind of combines all of the knowledge that you already saw, this diagram, that during the reprogramming process, all of the cellular hallmarks that we observe are rejuvenated and ameliorated. And of course, the next logical question was that, can we do this in vivo? Can we actually use the power of reprogramming in vivo to rejuvenate cells? And the first attempt was not really successful. I mean, it was successful because it worked, like in vivo reprogramming worked, but unfortunately the mice died. So from that standpoint, it showed that it's possible to reprogram cells in vivo, but in this first iteration, they let reprogramming commence fully, and that killed the mice by, within just 2 weeks because they got liver failure, organ failure, and of course, even the title of the paper was very gloomy. It said, reprogramming in vivo produces teratomas and iPS cells with totipotent features. Basically, teratomas are these cancerous tumors that we really don't want as a consequence of reprogramming. And the breakthrough that showed that you can actually do this safely in vivo and effectively came in 2016 with Alejandro Ocampo and others from the Salk Institute under the guide of Juan Carlos Espiso Belmonte. And they showed that actually you can do this safely if you do it partially. So you have to keep reprogramming partial. You have to do a very short duration of reprogramming to avoid this pluripotency, to avoid cells actually going back up the Waddington landscape. Because in vivo, that's not something we want or can afford, because we have to let the cells stay as they are. We have to keep the skin cells the skin cell, the brain cells as a brain cell. Otherwise, they lose function, and you can die. Your liver failure can kill you very quickly. And moreover, they showed that by using this approach, this partial reprogramming approach, you can prolong lifespan in this particular fast-aging mouse model, progeric mouse model, by up to 50% if you compare it to the first control group. And their mice, even visually they looked younger, but more importantly based on biomarkers of aging, like listed here, senescent cells, DNA breaks, inflammation levels. Based on those biomarkers, the treated mice were younger than the untreated mice. And their tissue histology looked better, and many other biomarkers that they looked at, the mice treated with partial reprogramming looked better than untreated ones. Now, of course, the big question by now, after a lot of other groups have replicated similar results, that partial reprogramming does seem to rejuvenate tissues and even animals. The next question is, how does it work? How does partial reprogramming work? Unfortunately, we don't have an answer yet. We don't know how it works exactly. I mean, we know it works on the level of epigenetics, but how exactly that causes rejuvenation, we don't yet know. But we have some kind of ideas, and maybe we can formulate some hypotheses. And here's my attempt at formulating one hypothesis. And we have to look at what do the Yamanaka factors do. And I mean, we know that OCT4, SOX2, and NANA are key factors for maintaining stemness in pluripotent cells, embryonic stem cells. Also, we know that OCT4 and SOX2 are pioneer transcription factors in that they can access closed chromatin, they can unwind it and start expressing previously silenced genes. But what is mentioned less often, though, is that OCT4 and SOX2 are also the key factors in triggering this maternal to zygotic transition that happens during early embryogenesis. And this is when the maternal genome gets silenced and the embryo's own genome starts getting expressed. And this starts at the blastula stage and finishes in the gastrula after implantation. And so I keep coming back to Vadim's excellent paper that showed that embryonic epigenetic age is minimal, not right after fertilization, but actually at the gastrulation stage. And I can't help but wonder if the two processes might be connected, and maybe the same networks of genes that are responsible for rejuvenation that we see after embryogenesis that are triggered by the OCT4 and SOX2 during the maternal to zygotic transition could be also triggering some of the genes that can produce rejuvenation that we see during reprogramming. So this is, this is one hypothesis that I have, and it gives new meaning to this quote that Oliver already quoted, that it's gastrulation that's the most important time of your life. Okay, switching gears from like what could happen, how it could work, to actually what we observe, and we observe that partial reprogramming already rejuvenates the transcriptome. It rejuvenates the gene expression pattern that we see in the cells that undergo the partial reprogramming process. And so while the exact mechanism of how it happens might still be unclear, from a practical standpoint, from a drug development standpoint, I think we can already start to use these results and use these observations for creating therapies that we might not fully understand. And I think we still don't understand how aspirin works, but we still, we're okay with taking it. And I think in the context of partial reprogramming, we might not fully understand how exactly it works, but we can still try to create therapies that could be therapeutically beneficial. And yeah, and there have been several studies that show this rejuvenation at the transcriptomic level and other levels, basically showing that reprogramming induces a shift towards a more youthful pattern of gene expression, which is, for our purposes, basically the levels of expression of key genes that we see in cells that undergo partial reprogramming are closer to the younger pattern than they are to the original older pattern that the cells started with before reprogramming. And so I think this is, as I mentioned, very important practical result that we should seize upon and harness the rejuvenating power of reprogramming for therapeutic purposes. And that's what we're doing at YouthBio, and many other companies are doing as well. And yeah, also, it's not just the gene expression pattern, also physiologically. There's data that shows that partial reprogramming induces physiological improvements in the cells and tissues in vivo, in the tissues that undergo partial reprogramming. So physiological rejuvenation, which is probably more important, because gene expression is great, but at the end of the day, you want your cells to be performing and your tissues to be performing at a more youthful level. And this is another hypothesis why rejuvenation by partial reprogramming is possible at all. And the hypothesis goes because the reprogramming process is gradual, and it's gradual in both the rejuvenating aspect and the cell identity changing aspect of it. And so here we have the graph of kind of the two processes side by side, and if you put them kind of one underneath each other, you can potentially find this therapeutic window where you already have accomplished some rejuvenation, but you have not yet gone beyond the point of no return of the cell identity changes. So you can still maintain your cell identity, you can still remain a skin cell, but you already have accomplished some rejuvenation. And that's why partial reprogramming is potentially able to work at all, because once you stop reprogramming genes for expression, the cell retains its identity, but it has been rejuvenated by the process of reprogramming. And since the 2016 Ocampo paper, there have been over a dozen other experimental confirmations of their findings that reprogramming induces some degree of rejuvenation and also prolongs lifespan in several different mouse models, progeric mouse models, but also in wild-type mice. And I just want to show you some results that I find particularly compelling. So here's an interesting study, because it showed that even a single bout of partial reprogramming can prolong lifespan. And it's not a huge result in terms of the increase in lifespan, but I think it's an important result because it shows that the result can persist. So even a single administration of reprogramming can result in both in a progeric mouse model. So they have two mouse models that they use: the progeric mouse model, the fast aging mouse model, but also a wild type normal aging mouse model. And they showed that both can exhibit this prolongation of lifespan by partial reprogramming. Here's a study from David Sinclair. I'm sure you've all heard about this study from 2021, where they were able to use partial reprogramming to promote neuronal regeneration after injury and even restore vision in mice. And I think now they're building upon this as a therapy for glaucoma, if I remember correctly, that they're trying to get into clinical trials actually shortly. I like this study. It showed that partial reprogramming can improve muscle regeneration after injury. Wound healing, and I mean, for the interest of time, I won't go into too much detail, but there's, as I mentioned, over a dozen studies by now that showed various aspects of partial reprogramming that can produce various therapeutic benefits. This is wound healing. Partial reprogramming can improve wound healing. Partial reprogramming can slow spinal degeneration, which is, I think, yeah, also a very unpleasant aspect of aging that a lot of people unfortunately experience. My own father had to have back surgery because of spinal degeneration. Also, this study, I really like this study from the Belmonte group. There's a follow-up study from 2022 where they showed safety of reprogramming, because a lot of the skeptics of reprogramming say that, okay, it's great that you can, use it for just a few months at a time, but you're bound to run into problems if you keep expressing these pluripotency genes on a repeated basis for too long. You're gonna, at some point they're gonna cause some cancer or something bad. Well, in this study, they had this 10-month treatment period where they periodically were expressing reprogramming factors, and they saw no adverse effects. In fact, they saw benefits in terms of, as I mentioned, tissue rejuvenation. And so in this paper, they also reported tissue rejuvenation. But I think more importantly, this paper is important because it showed the safety of this approach. And actually, Ocampo et al. Themselves have shown that not only did they use progenitor mice, they also studied in the wild-type mice, in normal aging mice, that 35 weeks of repeated induction of Yamanaka factors were not associated with any teratomas or any other adverse effects. But in the Belmonte study, besides safety, they showed multiple benefits of partial reprogramming in the tissues of mice. So not only were these 10-month-long periods of partial reprogramming safe, they were also therapeutically efficacious and beneficial. And finally, a lot of the skeptics of partial reprogramming were saying, okay, we only saw life extension in progenitor mice, in fast-aging mice. Talk to me when you can prolong lifespan in a wild-type aging mouse model. Well, here's a recent preprint from Rejuvenate Bio in which they did exactly that. They took a wild-type mouse model, and using a gene therapy approach, they were able to show that partial reprogramming can extend the lifespan of these really old mice, because they actually administered the gene therapy at a very advanced age for a mouse, 124 weeks. And again, it's not a transgenic mouse model, it's just a regular mouse, and you have to deliver these reprogramming genes into an already formed animal, and so they use the AAV delivery method, and using that, they've been able to extend lifespan by, again, nothing groundbreaking, but it was still a positive life extension, which, as a proof of concept, shows that you can do this safely in wild-type animals, and moreover, you do it using a gene therapy approach. Because again, there's a lot of skeptics that say, gene therapy, you can't deliver it into enough number of cells in an already formed organism to produce a meaningful therapeutic benefit. Well, I think this paper shows that you actually can. And we just have to build upon this to get more and more effective into targeting different tissues. And so this kind of is a nice segue to my final slide, the future directions. I think we have to study different aspects of reprogramming. And I think by now we already know that partial reprogramming really needs to be kind of tissue specific. Some tissues have to be avoided. As I mentioned, the liver is probably the tissue you really want to avoid because it's so amenable to reprogramming and can actually kill the mice. But also in terms of different combinations of factors, you have to find the tailored factors for a particular cell type. And so I won't, again, go into, in the interest of time, into the future directions too deeply, but there's, I think, a lot of the things that we need to explore collectively as the field of partial reprogramming. And I'm very optimistic that once we're able to accomplish these things, we'll have therapies that initially will produce meaningful therapeutic benefits in the context of existing diseases, and this will get us to FDA approval, get us on the market. And so then, as future steps, we can start, kind of build, combinatorial therapies that target several tissues and then can be used in healthy people as a preventative therapy that can rejuvenate a healthy person, bring down their biological age, and thereby, hopefully slow down their aging and maybe eventually even reverse aging. And so, okay, with that, I thank you very much for your attention. And if you have any questions, and hopefully I might still have 5 minutes for questions, would love to answer them. Thank you.
Thank you, Yuri. Yeah, we have a few minutes for questions. Does anybody have them? Yep, back here. few factors and they're intrinsically encoded, why hasn't evolution just turned these on whenever resources are abundant? Why are these shut down? What's the evolutionary reason why these aren't just active in keeping all organisms kind of longer lived?
Well, evolution doesn't want us longer lived. Evolution obviously wants us dead. So, and again, you know, you can have, I think, different schools of thought about the evolutionary benefits of aging, or is aging an adaptation, or just a kind of unfortunate thing that evolution chose not to deal with, or does evolution actually benefit from a turnover of generations? So yeah, and you can have its own conference on the different concepts of aging. So, but in some animals, like for example in jellyfish, actually they can reactivate essentially, or go back into embryo or polyp, and evolution was able to find this. But I guess other animals, evolution actually kind of doesn't want them to be immortal, or doesn't want them to live longer than it's optimal for their particular ecological niche. So that's at least my view.
Hello.
Oh, sorry, go ahead.
Yeah, I just wanted to ask you, are there any human trials underway, and are you recruiting more trials, let's see, in some other countries where maybe the laws might allow?
Right, so not yet, but there are some companies that are close to potentially close to clinical trials. One of them was David Sinclair's YouthBio, at least kind of from what was publicly available. I think they're looking into some eye indications. For some reason, I think glaucoma, but they might be targeting something else. And other, I think companies are close to clinical trials in some existing diseases, particularly maybe with hematopoietic stem cells or CAR T therapies that might use reprogramming, partial reprogramming to rejuvenate those cells. So we might see something in the clinic, you know, within the next 3 or 4 years, but not yet. But in terms of using other jurisdictions, definitely it's on the mind of developers, especially within longevity. Prospera is one of these kind of pioneering jurisdictions, as is, you know, Colombia and Mexico. They've been trying, say, telomerase gene therapies or clotho gene therapies. So it's possible, and maybe eventually even partial reprogramming will use one of those jurisdictions for its clinical trials, but not yet.
We have time for one more question.
Okay. Hi, this is Ene from New. I have a question. Just maybe if you could speculate in terms of, you know, this partial and yet systemic approach to reprogramming on epigenetic level, right? So we know that epigenetic changes very, very differently depending on the tissue, on the organ, and so on. So it's not the same rate. It doesn't really change the same way, etc. So can you speculate on what would be the appropriate, let's say, vectors to use in the future, like when and where we will be exploring the field to really approach that systemically? Because the same way as we're doing with gene therapies, I mean, you can squirt something into the eye, you have plenty of vectors, but systemically it's very, very difficult. So we're not there yet, but maybe if you could just kind of speculate where we think we might be going.
Yeah, absolutely, and I think you nailed it, and this is essentially, you know, the paradigm that we're going with at YouthBio, where we're using tissue-specific approaches for specific cell types, and so eventually the way we see long-term future of partial reprogramming for healthy people that need multiple tissues targeted is that we'll use targeted combinations of factors, potentially delivery vehicles, you know, with preferential tropism to that particular cell type or particular organ that we'd like to target. And so eventually it'll be a combination of therapies that also might have different inductor molecules, because right now you have to induce the expression of partial reprogramming. So, but first, you know, to get there and not to try to boil the ocean, you just use a particular cell type or a particular organ and in the context of existing diseases like, for example, Alzheimer's and the brain, and you target that, and if you can provide a therapeutic benefit using partial reprogramming and get that therapy approved, and you can then, build upon that, and eventually if you have, gene therapy approved for Alzheimer's, you can then use it off-label, and maybe even long-term use it for healthy people as a preventative therapy, of course, probably not the brain, but some other tissues like, for example, skin rejuvenation. You can start with wound healing, but eventually you can use it for cosmetic applications, for example. And so this is kind of the way I see, the step-by-step of getting there. Start with, particular one tissue, but then eventually get into combinatorial therapy. All right, I guess, yeah, that's it in terms of time. So thank you again. Thank you very much.